and welcome to Medisch Kompas, the medical podcast of the Netherlands, where we discuss topics on biomedicine, innovation, and research. I'm Christiana Costa.
Yeah, and I'm Henry.
And today we're with scientist Dave Speijer.
Nicely pronounced.
Perfect. Speijer is a scientist, lecturer, and molecular evolution, eukaryogenesis, and mitochondria enthusiast. He studied a lot of different things until he ended up to delving into the beautiful world of biomedicine.
Yeah, biomedicine or biochemistry. Yeah. I'm really just very interested in how cells work, especially if they make a part of a very complex organism like a human being. And there, of course, you straight away delve into all the medical implications of basic molecular biology and basic cellular biology and biochemistry. Now, I know that most people, when they hear the word biochemistry, they think, okay, I need to know something about it before I can study medicine. But is it really important? Yes, it is. It's the absolute fundament. And the interesting thing is, if you then delve into how all this evolved, you start to understand a lot of the things we are confronted with in general medical health. So I know that you, for instance, are interested in all kinds of diseases, but you also know as medical professionals or professionals to be that a lot of extra gain, you could say, could be from healthier lifestyles. That could be even much more important than all the other medical advances. And if you understand something about basic biochemistry and molecular biology, then you'll see, these lifestyle changes are really an important part of getting people into a happier state.
And there's one specific organelle that is like very important in this process.
Precisely. So if we look at eukaryotic cells, the cells that make up, for instance, human beings, and you could say all the more complicated cells, I'm not knocking archaea and bacteria here, so the prokaryotes. They are really biochemically very versatile. But to make up a very complicated structure, you need a eukaryotic cell. So why is this? That's already an interesting question. Why do eukaryotic cells allow so much innovation and so much complexity, while with all our love for prokaryotes, brilliant tiny machines, but it's always a brilliant tiny machine. So what has happened in this revolution, you could say, that suddenly opened up this enormous amount of extra complexity? Whatever you think about human intelligence, the human brain is a fantastic thing that does things that you can hardly conceive. And only a eukaryotic organism could do that. And then we come to the final reason why that is true. And that's indeed my favorite organelle, the mitochondrion. So, and if we delve a little bit into what the mitochondrion does, although it does an enormous amount of different things, it has been named, I think, in a Scientific American article in 1958 or something. Then suddenly somebody came up with this nice description that now everybody knows. It's the powerhouse of the cell.
Infamous.
Infamous. And also very incomplete. But it still stresses something that's really important for eukaryotic cells. And that's the amount of ATP they can generate. So the amount of energy they can make, and that's really superior when you compare that to prokaryotes like archaea or bacteria. Now, you can already imagine, okay, so this is fantastic. Now suddenly we have enormous amount of ATP. So I'm going to ask you some questions as well. And if I say it's the powerhouse of the cell, so why is that true? What do you know about this? What is so fantastic and what gives us these enormous amounts of ATP from mitochondria?
The electron transport chain.
Precisely. So it's the electron transport chain. And now you also understand where this name mitochondrion comes from. So it's a contraction of a particle with stripes, you could say. And these are the membranes that you see inside the organelle. It's full of membranes inside an organelle. And that's to increase the total surface. And if you've got way more surface, then you are able to stud that whole surface. And actually, we're talking about the mitochondrial inner membrane here. So there's an outer membrane and an inner membrane. And if you stuff that with inner membrane, and that inner membrane is then full of proteins, if you look at the outer membrane, it's like the normal membrane surrounding a cell. It's about 1:1 when you look at phospholipids or other lipids and proteins. So just mass, 1:1. But in the case of the inner membrane of the mitochondrion, it's 3:1. So there's way more protein than just lipids. Well, just lipids. I'm not going into that, but it's a really complicated structure. There are extra lipids for mitochondria like cardiolipin, etc., etc. Let's skip that. So why is it so chock-a-block full with proteins? And is the mitochondrion itself, because of these internal membranes, also chock-a-block full with these kind of proteins? That's because these proteins are involved in the very efficient generation of ATP. So there are people that, for instance, say you could never get a really big cell just being a prokaryote, because there the proton motor force is made by the membrane on the outside. And I don't have to tell you about volumes and surfaces. If I make a very big prokaryotic cell, I get a tiny increase of proton motive force, and that's what's going on here, for a larger and larger volume. But if you start just allowing these membranes to stack upon each other on the inside, then you can grow to a larger volume. And that's probably the most important reason why eukaryotic cells are so much bigger than prokaryotic cells, and also much more efficient. Now, I've left out one thing, so I'm going to tease you a little bit. This seems a brilliant invention. Now, we, as people that have lived already quite a while on this planet, know you don't get anything for free. You always have to pay. So, what could go wrong in this brilliant setup? Any thoughts?
What could go wrong in the setup? From an evolutionary perspective, I mean, you could think this is a kind of symbiosis, that because
it has to be nicely attuned.
Yeah, exactly. Yeah. So I do see some positive things. Maybe evolutionary, the bad things got kicked out of the mitochondria.
Interesting thing. Actually, one of the things you can't kick out, but you can adjust. And now I'm getting to the trade-off. So I've misled you a little bit. I've only pointed out, there's this wonderful internal surface extension and you've got membranes all the way. But all the membranes in the world wouldn't help you if the final acceptor in the electron transport chain wasn't the most efficient and optimal one that's around. So if you have the most electronegative final acceptor, then you can make way more ATP than just using, for instance, a hydrogenosome, where you end up making your electrons into hydrogen. That's electrochemically speaking, looking at the thermodynamics, that doesn't give you much ATP.
Okay.
But if you use oxygen, whoa, that really makes all the difference.
Yeah.
So it's internal membrane extension and using oxygen as the final acceptor. This is one of the reasons why most people agree, but we have to be a little bit careful. And maybe I should explain to your listeners that I'm nicely mixing up things that everybody agrees about and things that I think. And maybe I should point out where it's a personal opinion and not commonly accepted knowledge. So most people now agree, hey, prokaryotes probably around from 4 billion years ago. As soon as the Earth cooled off, life evolved and we got Luca, the last universal common ancestor. But if you look at the Leica, the last eukaryotic common ancestor, that's 1.8 billion years ago. So it took more than 2 billion years to come up with this invention. And one of the reasons why this probably can be explained nicely is this. I just said that you need oxygen as the final acceptor. Well, in the beginning, the Earth didn't have oxygen. That was you could say, chemically toxic byproduct of cyanobacteria that use sunlight. All this, photosynthesis. What is one of the waste products of photosynthesis? Oxygen. And now I have to be careful. It's, I think, 2.3 billion years ago. So 2.3 billion years ago, you have the great oxidation event. And that means that levels of oxygen in the atmosphere and everywhere were so high that certain metals like iron, etc., started to precipitate out. And you can see that nicely in bendings around these times. So we know that oxygen became more and more available, toxic to a lot of organisms, because it's a really reactive molecule, but a source of delight and ATP for bacteria that could use this as the final acceptor. And these are the alpha proteobacteria. And our mitochondria are the descendants of an alpha proteobacterium-like organism. By the way, this really is accepted. That's not my personal opinion. Everybody knows it. The mitochondrion still has its own tiny genome derived from the original bacterium. Okay, so this is all good. We use oxygen. We make, can I say this, shitloads of ATP.
Of course you can.
And so everything seems to be nice. But I just mentioned that molecular oxygen, it's relatively stable, but it's still enormously electronegative. It really wants to have these electrons and make water or whatever, or compounds. And that means that the way the respiratory chain has to be organized is really to be very, very careful about not losing your electrons on the way to the final acceptor before that should actually happen, because then you get a kind of unregulated uptake of electrons by oxygen. And this, as medically educated people, then you get reactive oxygen species. And ROS is the price we pay. And ROS, and that means mitochondria, have been implicated in, for instance, why we get older, in cancer, in all kinds of diseases. Now you, Christiana, said something very, very cool. You said, so it's a trade-off, and then the cell has to adjust to certain aspects of how the mitochondrion functions. And actually that's enormously important. Eukaryotic cells are full of adaptations to deal with ROS. And that leads to all kinds of stunning and very interesting and medically highly relevant aspects. So let's delve into them and see. And by the way, ROS, because it always is part of this system as it works, even if everything works nicely, you still have a tiny amount of ROS. That means that ROS is also a very important signal molecule. So in a lot of differentiation states, in immunology, you encounter it everywhere. So I've got a nice question that is related to this. We know that if we start moving around a lot, you use your muscles and you generate incredible amounts of ATP, and that means that your mitochondria are working like crazy. So they're using oxygen, and I also mentioned, whenever you do this, part of this whole process is that you also make ROS. Now, shouldn't it then be the case that if you are an athlete, that's very unhealthy? And that all these nutcases that say you should at least move around for half an hour every day, whether on bike or just... and by the way, that's shown that that's really healthy. It's one of the best things you can do. Just move around at least half an hour every day. Shouldn't that be impossible? Because you're making ROS. And ROS is a very dangerous molecule that can make mutations in DNA and oxidize lipids and mess up your proteins. So what's going on? Why is it healthy if you make these kind of amounts of ROS?
Adaptation.
Yes, yes. And there's a nice expression for this. This process is called mitochondria.
Yeah.
You remember that one?
Yeah, I was going to say hormonasis, but then I was like,
Hey, by the way, this is interesting. I also mess up my words quite a lot. And there are so many of them, and they resemble each other a little bit, which is also like the famous one where people mess up glycogen and glucagon. Oh yeah, you do that. That's. By the way, don't feel.
I find that
people
don't understand what I'm saying most of the time, so I just blend words out when I don't really know. So.
No, but this is an obvious one. It's a completely different kind of molecule with different function, but it's straightforward that this happens, especially when you're learning about these. Okay, so let's return to mitochondria. So what is actually going on? Actually, because the eukaryotes co-evolved with using oxygen, they had to adapt to endogenous formation of reactive oxygen species. Because this is something else that most people forget. If you look at the bacterium, the alpha-proteobacterial ancestor also used oxygen very efficiently. So why didn't it have these kind of difficulties?
I'm not sure. I don't know a lot about bacteria, but I guess they didn't make them endogenously. They just picked it out from the environment.
Well, the interesting thing is you pick up your oxygen and now part of the ROS is on the outside of the cell. You could also say, well, maybe it's even a defense mechanism. But the alpha-proteobacterium that was taken up is in the middle of the organism. So all ROS is on the inside. That's a different kind of situation already that needs extra adaptation. By the way, these alpha-proteobacteria also had ROS inside, because that's logical. Yeah. And they also had certain antioxidant enzymes. Let's do a little clinical sidestep. You know this terrible motor neuron disease, amyotrophic lateral sclerosis, ALS. Mm-hmm. It's sometimes called Lou Gehrig's disease. Horrible. In about 2 years, you completely, it's like your muscles dissolve. It's you can't breathe anymore and die in a rotten way. Now, this is a very complicated disease with a lot of different etiologies and contributions. But we know that in a small subset, there are mutations in the so-called superoxide dismutase gene. So what kind of enzyme is this? It's one of the antioxidant enzymes. So you don't take care in your motor neurons, in the specific ones that are targeted in ALS, and they die off quicker. And that's why you get this terrible disease.
Maybe I missed it, but so then to the question of why do we not have extra damage from then exercising, it's because there's also upregulation of the antioxidant enzymes at the same time.
That's it. If you don't do that, and suddenly you are making a lot of oxygen, reactive oxygen species due to disease, for instance, that's also an unhealthy situation in which the mitochondria need to generate more energy. Your immune system, for instance, does it. I'm sitting over here, by the way, for the listeners, as a person that's approaching 70, and I'm looking at two way younger people. Now, I know that the amount of oxidative damage in my body is incredibly high when you compare it to the.
Well, I really love the sun, and that's my only advice.
That's
very bad.
Very bad. Take care of factor 50 or something. So that also means that when we look at your genetic build-up, probably you're a little bit better prepared than if I would go out in the sun. So
that's very kind of you not to point to me.
Hey, you're English, so you suffer in so many different ways. Couldn't resist. By the way, listeners, I'm actually a great fan of English literature and English culture, etc. So let's get back to this part where we said, okay, so is it possible to stress this to such an extent? Yes, but interestingly, that's not only when you have way too much ROS from too much oxygen, There are also situations where you don't have enough oxygen because of a blockade. And then, interestingly enough, you could say, so there's less acceptor, so less ROS formation. But there's still an amount of oxygen present. And what's going on is that your normal electron flow is halted. You can't get to cytochrome C oxidase, which is the terminal enzyme of the respiratory chain. And that means that all these electrons are stuck and that's also a very dangerous situation.
Mm-hmm.
And this is very nice that you now give me the chance to really, you could say, almost define mitohormesis, because it's the optimal amount of oxygen and flow where the amount of ROS you make is normal and it just keeps the system on its toes. But with way too little oxygen or way too much oxidative phosphorylation, you get higher amounts of ROS. So in both cases, so this kind of stress, where you have not enough oxygen, also gives you oxidative damage, etc. So it's really a finely balanced system, which actually you could say is what life is all about. It's the tiny equilibrium between, life and death. And as soon as you drop out of a certain tiny range, oh presto, it's...
you're in the hospital.
You're in the hospital, or even worse, you're in the morgue. But okay, let's keep it fresh and light.
Yeah.
So
I have a small question here. So when we talk about myocardial infarction, then we learn in medical school, when there's hypoxia, then basically the mitochondrial membrane becomes more permeable, and then caspase and not caspase, how was that called? Cytochrome C?
Yes, cytochrome C. And no cytochrome C is released from...
And then, so that diffuses inside the cell, and then that activates the intrinsic pathway, and then you have the apoptosis things. And then you also said that hypoxia causes more ROS. I always thought like hypoxia causes some transcription factors.
Also, but I already mentioned this is enormously complex. The amount of... But one of the things that the cell has to do is it recognizes there's a lack of oxygen. Mm-hmm. That means you have to adapt your metabolism. And there is, for instance, HIF, it's hypoxia inducible factor. And that's one of these transcription factors that, and there are links to, for instance, all kinds of oncogenic processes here. Mm-hmm. The von Hippel-Lindau factor associates with this hypoxia-inducible factor. And if there are things wrong with that pathway, you get deregulated transcription and oncogenic processes. So there are all these crosstalks of, hey, is there enough oxygen for normal metabolic function? Actually, this illustrates that mitochondria are involved in everything. You mentioned apoptosis. Mm-hmm. So how does that role of mitochondria in apoptosis, in cell death, for listeners that are not aware that apoptosis means cell death? This is a controlled process where it's better for a cell to die, for the organism as a whole. And in the case of, for instance, low oxygen, that could be a good idea. So how does this mitochondrial know, I have to signal to the cell that things have gone wrong? Now, you mentioned that if you have way less oxygen, that things start to change with the membrane potential over the inner membrane, the source of all the ATP. And one of the reasons that this deregulates is that one of these electron carriers, so it was an electron transport chain, the one between complex 3, which is called cytochrome C reductase. So the other one probably is the cytochrome C oxidase. Okay. That means that there is a carrier in between. It's called cytochrome C. By the way, other carriers are inside, like ubiquinon, inside the membrane, but this is a protein. This is an electron carrier, one electron at a time, on the outside of the inner membrane. Oh, nicely complicated, on the outside of the inner membrane, in the intermembrane space. But nicely attached, you could say, via all kinds of non-covalent bonds to the membrane, so that it can play this role of electron transport. But if things go wrong with the membrane potential in whatever way, then the cytochrome C detaches it can go outside via the porins in the outer membrane and end up in the cytoplasm. And cytochrome C in the cytoplasm means, things are really wrong. And this can activate caspases. So these are proteases that start cutting up all kinds of other things. And if you start cutting up a lot of things on the inside of a cell, it dies off pretty quickly. So you nicely showed another very important function of mitochondria, their role in apoptosis and regulating that things stay under control. Okay, the cell dies off, but not via a necrotic process with all kinds of other messy things going on. And what this illustrates is that actually, metabolically speaking, I always dislike that people say it's an organelle. Yes, it's an organelle, but it's almost, metabolically speaking, the nucleus of the cell. Metabolically speaking, it's so important.
Yeah. Is this the new catchline over the powerhouse of the cell?
I like this one, actually.
Mitochondria is the nucleus
people,
of the
if
cell.
you want to quote me, forget about copyrights. I like it, so use it. It's like, metabolically speaking, it's the nucleus of the cell.
Yeah.
And that has also to do with another very important role, and that's the citric acid cycle, which is linked, of course, to the respiratory chain. And that has to do with all kinds of other very interesting clinical aspects, like immunometabolism.
Yeah.
And that immune cells are regulated. Shall we make it a little bit more complicated?
Yeah, definitely.
Do you like macrophages?
Oh, yeah.
You have to love them.
Dream of them. I dream of them.
So these macrophages are, you could say, the first line of defense when things go wrong and you get bacterial infection. And it's of course linked with the adaptive immune system, but it's more the major player in the innate, the first line of defense. By the way, this is way too naively put, but this is the way most people think about it. Now, if you look at T cells and B cells and macrophages, we used to think, okay, there is a B cell, there is a T cell, and there's a macrophage. Woo! As usual, we're so naive. There are many subtypes, different kind of roles. And with T cells, B cells, and macrophages, you could say one of the things that you could first separate them into is two classes: aggressive ones that take care of business, and the repair ones that say, hey, but now stop, you're overdoing it, and you're damaging way too much. And in the case of macrophages, that's also true. And these are called the M1 and the M2. Now, if you look at their metabolism and the role of mitochondria, really different. So in the M1, and now I have to be careful because I always mess up the two, but I think the M1, so that's the more aggressive one, has a so-called interrupted citric acid cycle. And that means that because the citric acid cycle and the respiratory chain are linked to each other, it doesn't give so many electrons to the transport chain. So things are really different. You could imagine that you get more ROS, other kinds of interleukins and other kinds of signal molecules are made than in the case of the M2, where it's like normal use of the respiratory chain. You have beta oxidation, you have so that means that you make an enormous amount of ATP. The citric acid cycle is a real cycle, not interrupted, and that intermediates of the citric acid cycle are used for other kinds of processes. So again, the mitochondrion is crucial. And that also means that we now are looking at medicine where you interfere with how the TCA cycle functions and how the respiratory chain function to modulate how macrophages work in certain kinds of diseases. Yeah. So there's an enormous amount of potential. I'm apologizing because it became already really complicated, but that's what biochemistry and life is like. It's really layer on layer on layer on layer. But it tells you again that, my favorite organelle and I'm going to use the new description, the metabolic nucleus of the cell is really of tremendous importance.
Yeah.
And all because of, you could say, an accident about 1.8 billion years ago. But I'm not going into eukaryogenesis because that's one of my other hobby horses. And there people would probably not all agree with my ideas. They're wrong. No. So this was a little field trip into mitochondria and how it works.
Yeah.
And
if I could summarize it a little bit, the previous one about the macrophages, I thought it was fascinating. So what I learned is that, so M1, you have a more disrupted, like the citric acid cycle and more like a chaotic, more raw species.
Well, chaotic, it's a program that the cell uses to then, you came with this nice example of the hypoxia-inducible factor.
Mm-hmm.
And that started a whole program of different genes, and then you get a different kind of response. So in this case, what actually is happening is if this happens to the TCA cycle, other kind of transcription factors are upregulated, and a whole other program.
Mm-hmm.
And that's the program of aggressive macrophages. So it's a kind of link. So it's not, it becomes a mess, it becomes another kind of program.
Yeah.
Okay.
Yeah. Does that mean that you could also target these kind of cells differently? Because now we have this idea in immunotherapy, at least for me, that you have the cluster differentiation factors that, like the proteins on the membranes that you target primarily. But since they have a different pathway they're following, they're different source in life, path in life, you can target those transcription factors or different.
Yes, and we also, the more we learned about how immune cells function, and this has less to do with mitochondria, but I think it's a very interesting and highly clinically relevant. Do you know about the cell cycle inhibitors that contribute to a much more positive prognosis in quite a lot of cancers? No? This is really interesting. So what people noticed was, I think the Japanese guy that Honsho got a Nobel Prize, and Allison or something, an American researcher, they both got a Nobel Prize for their findings and contributions to the so-called cell cycle inhibitor pathway in immune cells. Now, what has this to do with more efficient cancer treatment? Okay, so here we go. Our immune cells don't only recognize things from the outside, like viruses and bacteria. They also recognize, hey, one of our own cells is misbehaving. And one of the things that a misbehaving cell, like a cancer cell, can do, they upregulate certain pathways, they get deregulated, and you get certain proteins made that shouldn't be made by these kind of cells. And these can end up, for instance, in the so-called major histocompatibility classes. And that's how you recognize whether a cell is misbehaving or normal.
MHC for the medical students.
Yeah. So the MHCs. So and these are encoded in the genome by the so-called HLA genes, the human leukocyte antigens.
Very interesting.
Okay, so and these are recognized by T cell receptors, and you've got an enormous diversity, just like the diversity of antibodies. And if a T cell receptor encounters an MHC with a strange molecule and it fits, hey, I shouldn't recognize something, such a T cell can be activated. Now, an enormous amount of different things happen, but for instance, normally we never get Kaposi sarcoma. That's a very rare, but if you get AIDS, your immune system doesn't function properly and you suddenly start getting Kaposi sarcoma. And that tells you something very important, that your immune system takes care of a lot of cancerous cells that you're completely unaware of. You don't even know that it happens. It's just usual business. Now, the interesting thing is sometimes you do get cancer. So your immune system wasn't able to stop a cancerous development in its tracks. And what people found out was, and this was almost a little bit of luck that you don't only activate a T cell by having this interaction between the MHC and the T cell receptor. No, there are a few extra contacts that are needed as well before you get full activation. And this is because we talked about equilibria, about a very sensitive area where it still works or it doesn't. If your T cell is activated way too quickly, you could start destroying normal good cells, etc. So there's an extra layer of safety, you could say. Cancer cells, well, they quickly develop quite a lot of different mutations, etc. And sometimes they are nicely able to, for instance, induce one of the cell surface molecules that tells the T cell, no, nothing wrong here, nothing to see, everything is okay. I know your T cell receptor was activated, and these are the so-called
checkpoint inhibitors.
Precisely. They activate a checkpoint inhibitor in the T cell. And then the T cell says, no, I'm not going to divide. I'm inhibited at a certain checkpoint, blah, 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 blah. So if you recognize that this is a very important thing, then you know, but if I could interfere with this stop signal, my anti-cancer treatment would be way better. Because now the T cell still is activated and the immune system can be used to destroy the tumor. And that has made an enormous contribution to the survival rate of certain types of tumor. So you just bring in, for instance, and now I'm having to be careful because maybe there are already other ways of interfering with it, but an antibody that recognizes this cell surface molecule. And then, there's no interaction anymore because it can't attach itself to the ligand on the T cell membrane.
We speak briefly about this in an episode coming soon about rituximab and these for rheumatoid arthritis specifically.
So, it's not only in cancer, but in a lot of autoimmune diseases, etc., etc. There you could use them, for instance, to inhibit the T cell because their T cells and B cells are going berserk and attacking normal tissue. So it's playing around, but the more knowledge you have, the better things are. And it's still pretty tough when you're diagnosed with cancer, but there are now many cancerous diseases where it's just, with these kind of medications, you've got a pretty good prognosis, while it used to be a death sentence 15 years ago. So real progress is being made there. By the way, you see that I'm also interested in things where the mitochondrion doesn't play a completely dominant role. So there are side effects, this is an interesting one.
If we go back to the mitochondria, where we had this train of thought. So we started with the ROS, with the reactive oxygen species. We went to the example with the macrophages, which was very nice. And we started off also with the electrotransport chain that is connected to the TCA cycle. And then I also know that, part of the TCA cycle are also signaling molecules, like these things. What are the implications? Because I think I read something also about cancer and these kind of...
Yes, that's true. Well, first of all, if the mitochondrion is really so important and it has its own tiny genome, why don't we have many more? There are mitochondrial diseases where the mitochondrial genome or proteins that are imported into the mitochondrion but nuclearly encoded. Why don't we have many more of these kind of diseases? That probably has to do with the fact that the quality control of mitochondria during embryogenesis is rather high. It's probably one of the reasons that I think I read somewhere about 30% of pregnancies end up in a abortion early on, hardly noticed. And that could be due to the quality control of mitochondrial function. It's so essential that as soon as there are tiny mistakes, you don't go through the hoops of embryogenesis.
I imagine if it ever did go wrong, also there'd be quite a hard selection drive away from it being. It'd be quite dangerous to have.
Precisely. You won't have many children. So there are. And that has to do with what you talked about, that it not only generates the amount of ATP, but it's also via the TCA cycle and the functioning of these kind of aspects of mitochondrial metabolism, signaling quite a lot. And intermediates of the TCA cycle, just like cytochrome C was an intermediate, you could say, of the respiratory chain, can go out mostly in a slightly modified form. So not the actual TCA cycle intermediate, but these can indeed signal. That's what happened, for instance, in these M1 macrophages. And so the halting point where it wasn't a complete TCA cycle, something happens to that intermediate. And that's then influencing transcription factors and sometimes directly. If you, for instance, you know how the chromosomes are modified by acetylation and these acetylation groups, so these acetyl groups, they can come from the mitochondrion. So there's a really strict regulation by the mitochondrion of what happens in the cell. So signal molecules are essential. And the TCA cycle is of enormous importance in all that. So you're absolutely right. That's one of the things how that works.
Could that mean that supplementation of specific molecules or even diet can have an effect?
This is a really good one, because this is one of the hobby horses of the health nuts.
Yeah.
and for instance, saying, okay, you one of these electron carriers in the transport chain, as it's called a vitamin, which is not, but it's ubiquinone, Q10. You see that in the skincare. All kinds.
I actually genuinely put that on about 15 minutes before.
Every day.
Okay, so and these are so nicely balanced because they're so essential that bringing Q10 from the outside, well, I don't trust this so much. So what is true is that under certain circumstances, things like vitamins or antioxidants, because a lot of the vitamins are antioxidants as well. So not as a protein, but for instance, superoxide dismutase, the one I talked about in ALS, they need cofactors. And these cofactors are electron sinks. And in a lot of these pathways, the vitamins pop up. That you see, but the final or almost final acceptor of your electron is vitamin C or vitamin B12 or etc. So a lot of these vitamins are temporary electron carrier, just like ubiquinol. But in most cases with normal, healthy eating patterns, all that stuff is not necessary. There are a few exceptions when you're pregnant and you need somewhat more. You know the standard ones. And maybe when we become older, there is the slight possibility that a combination of vitamin C and vitamin D, and then one of the specific ones, is a little bit healthy to lower the chance of getting cancer. But these things are, I'm not really convinced, but there are indications, some. But
I was thinking more of like somebody has cancer and you want to change the microenvironment that the cancer is in. So you want to maybe inject some molecule around it or...
Okay, so, well, you could say that the cancer itself is also changing its environment in such a way that's better for it and allowing metastasis, I should say. See what happens? You grow older and you start making more and more mistakes, etc.
Yeah.
But now, this is, by the way, interesting because am I allowed a little detour where the mitochondria comes in again? Because you said, okay, so the environment of the tumor is different. And then I said, yeah, but the tumor itself starts changing its environment also. And now we come to something where the mitochondria again plays an enormous role. And I think your listeners will be interested because I'm going to use the term Warburg effect.
Oh, yeah.
Okay, so tell me about it. What is the Warburg effect?
Okay, that's quite long, but I remember glucose,
Yeah.
something. I have to retrieve it. And I also remember that too much glucose is not good because basically tumor cells are cells that proliferate very quickly. So they tend to accumulate glucose around them and use it for multiplication or something.
This has to do with part of it. But the interesting thing is that the Warburg effect was discovered with cancerous cells by Otto Warburg, a brilliant biochemist. And he noticed that if you have cancerous cells, they start doing something rather weird. So, let's say you give a normal cell glucose. What does it do? It uses the glucose, it makes it into pyruvate in the cytoplasm. That's called glycolysis.
Okay.
And then the pyruvate goes into the mitochondrial, and there you get 90% of the ATP, even more, made in all the things we discussed.
Mm-hmm.
So, if you don't have a mitochondrion, then glycolysis would stop. That sounds weird, but it has to do with the fact, or normally it would stop, with the fact that you generate a electron carrier that's full up with electrons, and that's NADH.
Yeah.
And the interesting thing, if you don't make NADH into NAD plus again, so listener, that means that 2 electrons are gone, NAD plus misses 2 electrons, NADH carries 2 electrons. And in glycolysis, you are making from NAD plus NADH. Now, let's say you end up with pyruvate. Yeah, you have to stop at a certain moment in time because all your NAD plus is chock-a-block full with electrons in the form of NADH, and you can't go on anymore because the first step needs the NAD plus. Now, normally that's no problem because the pyruvate goes into the mitochondria, and the NADH made in the cytoplasm goes into the mitochondrion as well. And there these electrons are taken care of by our favorite respiratory chain, giving us enormous amount of ATP. So that means that normal cellular behavior is using all the glucose and making it into CO2 and water and making ATP, 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 ATP. Now, this is normal cellular behavior. Now you have a cancerous cell, you give it glucose, and it does something weird. The pyruvate doesn't go into mitochondrion, it's formed into lactate. And you know what the side reaction is, or the coupled reaction? If you go from pyruvate to lactate, you use an NADH, and that becomes an NAD+. And that means that now glycolysis can still go on.
Yeah.
But you have a waste product called lactate. Mm-hmm. And that's enormously energy rich, and you just throw it out of the cell. That's weird. Why does a cancer cell do this? It throws away an enormous amount of ATP. It throws an enormous amount of building blocks, etc. Now, there has been an enormous amount of speculation why the Warburg effect occurs, not in all cancer cells, but in quite a lot. And by the way, you could say that what we encountered in our macrophage M1 resembles the Warburg effect a little bit. You hold the TCA cycle, you don't have the normal respiratory chain, and you also end up with building blocks. And probably there's something we can learn about the Warburg effect here as well. So you could speculate for hours why the cancerous cell does this. First of all, you have to remember that this cancerous cell is inside a human body. Now, if it still was a single-celled organism, this would be a very bad choice because you throw away all your energy. But you don't have to deal with this hardship in a human body because blood glucose levels are about 5 millimolars if everything is normal and stable. So, hey, more than enough glucose and you just throw out your lactate, no problem. So it's not an energy constraint anymore. So why does it then do that? And here an enormous amount of speculation goes on. But one of the things that probably is going on is that if you do the complete glycolysis, then you use all the glucose and make it into CO2 and H2O. And that means that the TCA cycle can't be used to take its intermediates and make them into other stuff. Because one of the things, and that's why this is perfect, I haven't stressed enough, is so if you use your TCA cycle just to cycle, nothing is formed. You're cycling. And what goes in, an acetyl-CoA, that contains 2 carbon atoms, and these are thrown out during the TCA cycle. 2 CO2 go out. So there's no net change. It's oxaloacetate plus acetyl-CoA makes citrate, blah, blah, and all the way. But if you don't use these NADH and this whole normal cycle, then you can take out intermediates. And that happens quite a lot. Because the TCA cycle is not only central to catabolism, so making ATP, but there are also an enormous amount of intermediates for things that the cell really needs, like amino acids, like heme groups, like making glucose again from oxaloacetate. The liver, for instance, has gluconeogenesis. So that means these things are very tightly linked. And one of the things that you could think is, well, the cancer cell actually isn't so interested in making ATP because there's more than enough glucose. No, it wants to use the TCA cycle for other kinds of stuff, like making new molecules and dividing as quickly as possible. And for that you need new DNA, new proteins, etc. So that's one of the things you could say about why the Warburg effect occurs.
Yeah. I was also always thinking that one reason that the cell enters the TCA is to regenerate its NADP plus.
There is an exchange between NADH and NADPH. So that's, but that's really complicated.
Okay,
so that has to do with transhydrogenases, etc. Dear listener, if you're interested, just look up the word transhydrogenase. So, one other way of looking at why the Warburg effect occurs, and it's also interesting, I think, but this is more of a personal hobby horse of mine. Normal cellular functioning, just using pyruvate and beta oxidation to make ATP, and that uses a lot of oxygen. And that means that these kind of cells, the differentiated states, like for instance, a heart muscle cell, so a cardiomyocyte, that uses beta oxidation all the time, and that uses an enormous amount of oxygen because it has to make ATP all the time. Well, these kind of cells, because you use have a lot of antioxidant measures. They're checking whether everything is okay, whether it's not time for apoptosis, because so there's a lot of extra control. Now, there's one thing that a cancer cell doesn't like: extra control, where it's told, hey, stop dividing, commit suicide. And that means that also this shift allows a cancerous cell to make a mess of things without being directed into the apoptotic hole of oblivion, you could say. So there's also this kind of shift. And there is, you know that in cancer, quite a lot of the cells that become cancerous, certain tumor suppressors, and these are mostly control proteins, are mutated. And that's then not the mitochondrion, but you could say the mitochondrion is also a big, giant tumor suppressor.
Yeah.
Like the P53. So as soon as these kind of controls go out of the door, then you get cancerous development.
Yeah.
So that's you could say an overall general picture of interpreting what's going on.
Yeah.
And I don't know whether it's completely true, but I find it illuminating to think about it like this. I hope you're still illuminated as well.
I think it's fascinating. I do really want to ask one last question about this. But so we have a cancer cell that really uses a lot of glucose because there's vast of it. But what if we make sure the person starts a keto diet and then their blood glucose is like quite low? I know that's not feasible to have a very low blood glucose because, the brain needs energy.
Yeah.
But what if we try to deprive it from its glucose? And what happens to it?
Okay, so very interesting question. By the way, we could talk about this for another hour.
Yeah.
We won't. Dear listeners, don't say,
we'll be done.
please, no. But there are a few things that are really important here. First of all, if you look at a keto diet, whether a constant keto diet is really healthy, people are not quite sure, but probably it's healthier than just eating as much as possible from high glucose, high fat sources. So not bad. I think the chance that you get cancer when you have, for instance, a habit of intermittent fasting and temporary ketogenesis from your diet, that's all good. When you already have cancer, your idea of, let's deprive it of glucose, alas, is not feasible.
Mm-hmm.
And that has to do with the following aspect. Yes, you're right. You can induce the amount of ketone bodies. But there's one difficulty, and that's this. That normally you have a glucose level of about 4.5 millimolars, constant. Unless you have just eaten, then it goes up. Insulin takes care of that, and you return back to 5 millimolars. If things go wrong and your liver isn't able to generate glucose anymore after fasting for too long or just hunger, there is no source of energy, then you can get hypoglycemia, and that means below. And if you are around 2 millimolars, your brain really gets enormous difficulties and you go into a coma prior to dying. That's it. Even in a state of prolonged low glucose intake, where ketone bodies are upregulated, so ketogenesis occurs in your liver, then at the maximum situation, after 3 weeks of practical hunger, close to, but still being able to generate these ketone bodies, 50% of the energy of your brain is from ketone bodies. But that's the limit, you could say. 50% still has to come from glucose. So, and that means that the 4.5 millimolars of glucose is just obligatory.
Yeah.
Now, it's very interesting to think, but hey, my heart just uses fatty acid oxidation all the time. Why doesn't the brain do that? Why doesn't the brain use fatty acids then?
Blood-brain barrier?
That's what most people thought. But the interesting thing is we now know that there's quite a lot of fatty acids inside all kinds of cells. They can make it themselves, the neurons. Yeah. And we also know that actually fatty acid can cross the blood-brain barrier. Mm-hmm. So that's not the reason. Now, I'm not going to talk about this because this really is a hobby horse. I speculated about this in a 2010-2011 article, that this really tells us something very essential about eukaryogenesis, etc. I'm just going to say this. What's the difference between cardiomyocytes and muscle cells? There is quite a lot of regeneration. So the cell dies from oxidative damage. Well, you just make new muscle cells. That's not so easy in the heart, although there are indications that, but there it's not a big deal. And you can miss a few cardiomyocytes and your heart still will function. But what happens if neurons die off? And they do that all the time. They can't be replaced. And that means your memory is going, because actually a neuron is part of your memory. A good neuron is with you all your life. Well, not completely, but in principle, quite a lot. There's also redundancy, should be there, and then you won't get dementia when you're 30. So that means that actually you can't do certain metabolic things in neurons that generate an enormous amount of ATP, but that might give you too much ROS. Because the regenerative power of neurons is much lower than in other tissues. And you would lose your mind. That's it.
Mm-hmm.
Actually, dear listener, this is one of my hobby horses, and not all people agree with this kind of interpretation. But I think that this tells us something really important about the fundamental metabolic processes that occurred during the evolution of the first eukaryote.
Yeah.
So, and that we can still see this in differential behavior of our tissues, like the differences between a heart and a brain.
I think that's super fascinating.
Yeah. Well, maybe this hobby horse I can really talk about for a very long time.
Maybe next time.
Yeah. Okay. So then I'll leave the listener with one extra, you could say, important word, and that's peroxisomes.
Oh, yeah.
Look it up, because that has to do with this kind of functioning as well.
I remember Zellweger's disease.
Yeah. So, but the very important oldest pathway in peroxisomes is probably beta oxidation.
Cool.
So, and now you see certain links. So look it up. And I published on that quite a lot. So you can find these things in PubMed as well.
The backup favorite organelle next to the mitochondria. If we get bored of them.
It's sometimes called the little brother.
Oh, wow.
The little brother of the mitochondria.
Yeah. Well, about the mitochondria, then maybe it's nice to quickly summarize what we talked about. So we talked about the reactive oxygen species and about the intrinsic apoptotic pathway. We talked about the electron transport chain and how that is also coupled to the TCA cycle, and the intermediates of the TCA cycle can act like satellite molecules and act like messengers, and that maybe the program in which this whole process happens could be different in cells, let's say macrophages M1 and M2. We also delved a little bit into immunophysiology. That was very interesting. Maybe also about the checkpoint inhibitors, and about the fact that the mitochondrion also has quite some control, and there could be some like dysregulation. Also about the how do you call that again? The for the glucose in cancer.
The Warburg effect.
The Warburg effect.
Otto Warburg
effect. And some other clinical pearls.
It's sometimes called oxidative glycolysis, and that means you only do glycolysis while there's more than enough oxygen. Yeah. That's strange. But I don't like that way of describing it, because normal oxidative glycolysis for me is just going through everything using the TCA cycle, oxidative phosphorylation.
Yeah, exactly.
And giving CO2 and H2O at the end.
Yeah, because glycolysis
ATP,
always happens,
ATP, Precisely.
Exactly. But
I thought this is a perfect summary, by the way.
Yeah, perfect.
You could also said we talked about everything.
Yeah.
I have a final question, if I may. Yeah. If we a broader scope on this, a little bit out of the cell, but when you look at medical students, do you have advice for how they should approach biology to understand it fundamentally in regards to how it affects medicine?
Yeah, I do have one thing. I know that it's really a very complicated field. That's clear. And biochemistry and all the reactions, etc. It's really the finer details. It's a complicated field. And I also know that quite a lot of doctors to be, well, they're not fans to say. It's because it's not that it's not fascinating, but it's so complicated. Well, actually, the complications are sidetracking you. If you have an idea of, does it make sense? How does it work? What we try to do here in the discussion. Yeah. Oh, but then that kind of... So the details of the reactions and the name of the enzymes and all the specific intermediates, you can all look that up. Forget about it. That's not... But follow the reasoning. Yeah. Get a grasp of, so practically everything that happens here is reduction oxidation. So that's electron transport. The electrons go from this one to that one to that one. So if you use general concepts like that, then suddenly biochemistry starts to make sense.
Yeah.
You get a good feel for the overall picture, and the details, who cares?
And then we link that to the pathology and then
precisely.
disease. Yeah.
Precisely. And I think that's a good way. And then sometimes, not the happy few, but the people that are really infected for life, they suddenly see, but biochemistry is fascinating.
No.
It's so wonderful.
Very dangerous.
Very dangerous. So about 1 in 10, you'll destroy their future because they fall in love with biochemistry. That's what happened to me. Yeah, and now you can describe, and we are watching indeed a very old and
self-destructing,
precisely. So I think that's the best way to look at it. So get a feel for the concepts, and if you do that, it's so much easier to understand what's going on.
Yeah.
The details are less important.
Yeah, beautiful.
Okay, isn't that a good philosophical wrap-up?
Yeah, thank you so much for coming and speaking with us today.
It was my pleasure.